So the normal process is before you trial any drug, you first want to see whether it's plausible that this drug may have an effect. And based on the literature, so FDG is something that we use very commonly for what we call PET scans. So PET scans are scans in which you inject this drug FDG, and it goes to the area where there's a lot of inflammation in the body or there's a lot of activity going on in the body. Now, what they found was that in patients with COVID with pneumonitis, with the involvement of the lungs, the injection of the FDG led to a lot of activity seen in the lungs. So they therefore concluded that FDG reaches the lungs at the site of inflammation very avidly or very effectively. Now, this drug 2DG is supposed to be very similar to FDG. It has been trialed or it has been used in cancers, is my understanding. What it does is it somehow depletes the cells of the mechanisms that are needed to grow and multiply, which is why I think the hypothesis is that the 2DG will reach the lung cells, will deprive the virus of substrate or material that the virus needs to survive, and will therefore lead to the virus being killed or the virus dying. So that's the biological plausibility. That's where they got the idea from to use this particular drug. The study was designed to be used in patients with moderate to severe COVID. So patients who had low oxygen levels, patients who had a high respiratory rate, who had a severity score greater than 5 on the WHO scale. So it wasn't designed for people at home. It was designed for reasonably severe individuals. The press release mentions the fact that the drugs seem to work better in terms of reducing the oxygen consumption of individuals, reducing the length of stay or reducing the duration of illness. And these are very promising results. However, I would like to see the actual details of this in terms of what was used as a measure of outcome. Was it mortality that was used? Was mortality one of the things that they measured? Was severity of disease in terms of oxygen dependence one of the things they measured? And also, who was the person assessing? What were the objective criteria used to assess whether a person got better or worse? They did a phase 3 trial, which involved about 220 patients. Now, a few caveats need to be mentioned about this trial. So based on my understanding, based on what I see on the CTRI website, there was no blinding. So the person giving the drug, there was no placebo control, which means that the person giving the drug and the person receiving the drug both knew that they were part of the active arm of the trial. That's our understanding. Now, by virtue of that, a little bias gets introduced into a study. So the way a physician will assess that patient based on his or her knowledge that the person is receiving the active drug can actually change. So if I know that a person in a particular trial is getting the active drug, I would want to see positive changes in that patient because I believe in that drug, which is why I'm participating in the trial. And that can sometimes introduce a level of bias. Based on what's up at the CTRI website, the dose that is recommended is 90 milligrams per kg per day in 2 divided doses. And it's given in the oral form. So it needs for a person to be awake, alert, and able to consume oral drugs. So individuals who could not take drugs orally were excluded from the trial. I think a recurring theme that we've seen throughout the past year is that a lot of different drugs have shown promise in small trials. And the moment you test it in a rigorous manner, where you have a randomized study or you have a placebo-controlled study, or a really large study like these adaptive studies which have been done, such as Recovery, such as Solidarity, you've realized that a lot of drugs which look promising in small studies eventually do not pan out to be really effective for COVID-19. So I think that's the word of caution that we must all realize, and we must all exercise caution while interpreting data from small studies, unless the signal is so strong that it can't be ignored. So unless there's like a 50% mortality benefit or there's a huge signal, a small study has to be taken with a grain of salt, because just by statistical probability, you can get results in a small study which may not be true, which may not be replicated, which may not be extrapolated to a larger population.